Hi guys, this is Alicia with Good Morning Sunshine. So today I've only had 2 lectures, but I have client simulation coming up at 1, so I just thought I'd make a video real quick. So then I have to prep for that because I have to take out all my piercings and be in a white lab coat because we're apparently student doctors and we're going over history and stuff. But anyways, we had 2 classes still. We had pharmacology, so I'm going to start with that. We were on antibacterial agents for systemic therapy, so protein synthesis inhibitors. So all we're going to talk about today is the drugs acting on the 30S ribosomal unit. So those would be the aminoglycosides and the tetracyclines. So the aminoglycosides, drugs in this class are systemic. This includes gentamicin, amikacin, and tropomycin. They're also used intra-articularly and for RLP, not meaning regional limb perfusion. This is used a lot in horses for their joints and legs. So drugs in class that we're going over, we have neomycin. These are local drugs. Neomycin is often used for the eye, but can be used orally since it's enteritis in swine, so pigs. Also, streptomycin is usually used in food animals systemically. And then mechanism of action, this all having to do with protein synthesis. So bind to the specific receptor proteins on the 30S ribosomal subunit. It's irreversible binding, so it has a covalent bond. So the formation of non-functional proteins and adherent proteins are inserted into the cell wall, this leading to altered permeability and ultimately leading to cell death. So they're very bactericidal, these drugs. It is important because they're going to kill bacteria quickly. So usually used for very severe infections, so you get a rapid response that way. And then we have post-antibiotic effect. These drugs are known for having a post-antibiotic effect, alter proteins in the cell membrane, increase drug permeability, so can get into the cell walls very easily. Thus, this is why they kill bacteria very fast. The effects last long after the drug is gone from the plasma, so that's a plus as well. Spectrum of activity, gram-negative aerobic organisms also have activity against staphylococcal organisms. Includes some methicillin-resistant Staphylococcus aureus, also MRSP. It's ineffective though against streptococci and no activity against strict anaerobes. It also requires oxygen for transport into the cell. So for clinical use, severe gram-negative infections and also sepsis. Use may be limited by adverse effects though, however. It's often combined with the beta-lactams. Why? Because of synergism. So administration of a beta-lactam, then the administration of aminoglycoside. Again, remember we don't mix these in a syringe, but if we administer one and then administer the other, this is very good for aerobic coverage as well as anaerobic coverage, because remember, aminoglycosides cannot touch strict anaerobes, but beta-lactam can. So by using both, we get a complete coverage. So which one to use? So amikacin is more active. It's also less toxic and resistance is less likely to develop. However, amikacin is very, very expensive, so we usually just use gentamicin. So oral absorption is poor, so they don't make it across GI membranes. Neomycin, however, this can be given orally, so it's because it stays locally in the GI tract and treats bacterial infections that are locally just in the GI tract. IM absorption is almost 100% absorption, but IV is the most common route here. They're high water solubility, they have low protein binding, they're highly polar. So what does this tell you? It means they have a low volume of distribution. They stay in the plasma and the extracellular fluid. They do not penetrate into the CNS, so the central nervous system, or the eye, or the prostate. They won't treat abscesses either. So example being R. equi. This is an intercellular pathogen. It's sensitive to gentamicin. However, we used to give it in horses. This was because the culture that we did in lab, or that they did in lab, said that they could, but horses would then still ultimately die after giving gentamicin because they couldn't get into the infection which was inside the cell. It's excreted metabolized by the kidney, glomerular filtration. The half-life is very short in these. It's 1 to 2 hours if renal function is normal, but it's dosed once a day. Why? Because they have post-antibiotic effect that I talked about earlier. So the pharmacokinetic and the pharmacodynamic interaction, so it's concentration dependent, so also important as to why we only dose once a day. And there's reasons to dosing every once a day. We used to dose like 3 times a day, but that became a problem because of the adverse effects. So adverse effects is dose-dependent nephrotoxicity. This is from when we used to dose 3 times a day, like I was talking about. And then we have nephrotoxicity, renal toxicity, and it's bad for the kidneys. So the risks are greatly decreased when we just giving it every 24 hours. Once daily dosing benefits are we administer at a higher dose, so we reach higher maximum concentrations faster. Active after the drug has left the plasma, like I said, so that's the post-antibiotic effect. And then nephrotoxicity is dose dependent, so it's less likely to occur if the Cmin is less than 2 to 3 UG per mil. This is a concentration left over before we redose the medication, so after 24 hours when we give another dose. The drug has an active transporter on the renal tubular cells through an active transport mechanism, so don't get into intercellular concentrations in the bacteria, but do have active transporters on the renal tubular cells that allow uptake of drugs into the renal tubular cells. It can saturate mechanisms by giving higher doses, so a number of transporters limits uptake into cells. So by giving once a day, we have less drug inside the renal tubular cells, which is good, so it's safe to redose. If we give it more than once a day, we saw that we would end up exploding the cell because it has way too much drug inside the renal tubular cell at one time. Nephrotoxicity, gentamicin is the highest for this, then we have tobramycin, and then amikacin. Risk is increased with dehydration, so we don't administer this drug when the dog's dehydrated, they have fever, they have pre-existing renal disease, or and we don't give it to neonates. Toxicity is often reversible, so when you start noticing the creatine going up in the blood work, you should be concerned. So treatment is prolonged due to slow elimination of the drug from the kidney. Administration of calcium protective. This calcium actually competes with the active transporter on the renal tubular cells to prevent the aminoglycoside from being taken up. It also increases protein binding, so this will inactivate some of the drug also when you're reversing the drug. Other adverse effects are ototoxicity, the drug accumulation in the paralymph and the organ of the corti, which is in the ear. We have increased risk in CAS and increased risk with topical otic products. We have vestibular toxicity, which means the head tilt. I'm sure you guys have seen before. Ataxia, impaired writing reflexes, and collicular toxicity. So other adverse effects, we have neuromuscular blockade. This is the competitive interference with calcium transport at the motor end plate. Should not be administered concurrently with certain anesthetics, skeletal muscle relaxants. Should not be used in diseases of the NMJ, so this causes butilism. Considered teratogenic in humans, can impair formation of the kidneys, fetal rats in the first trimester, and it's not really investigated in other species. Drug interactions, so beta-lactams react very well within the body with these drugs. Bacteriostatic drugs, we don't want to co-administer with these. Other nephrotoxic drugs like NSAIDs can cause renal disease if the aminoglycoside is mixed with an NSAID as well. So they're taking both at the same time. This could cause renal disease. And then furosemide or other diuretics, unless the patient is well hydrated, increased risk of neurotoxicity, possibly due to the dehydrating effects. And then we have mechanisms of resistance. So number one is enzymatic modification, so mostly plasma mediated. Aminoglycosidases is which inactivate aminoglycosides. Aminocasin is the least affected. Lesser altered ribosome binding and lesser reduced uptake, so active transport. We do use them in food animals. It's not illegal, but if you give them any route that causes systemic absorptions, this includes IV and IM routes, then there's a 10-day milk withhold, but an 18-month meat withhold, so they'd just be out of production this way. And then we're going to go into tetracyclines. So their mechanism of action, it binds to the 30S ribozymal subunit. It blocks amino acids being added to the peptide chain, and it has the inhibition of protein synthesis. Binding is reversible here, and tetracyclines are bacteriostatic at all concentrations. So the drugs in this class are oxytetracycline, which is used in horses and food animals. We have chlorotetracycline, which is used in food animals. We have minocycline used in horses, dogs. It's most active here. And then doxycycline, of course, is horses, dogs, cats. So spectrum of activity is broad, but resistant is common. So gram-positive and gram-negative bacteria, aerobes and anaerobes, used for chlamydia, rickettsia, spirochetes, mycoplasma, L-form, so cell wall deficient bacteria, and then some protozoa. Clinical use, he's not asked for specific names, but for cats and dogs, we have all different things that we use. So like I said, Ehrlichia, rickettsia, and mycoplasma, Haemophilus, and in birds we have Chlamydophilia, Psittaceae, and in horses we have rickettsia, Ristia, Anaplasm, Phagocytophilium, and Losonoma intercellularis. For food animals, they're injectable. Extended release indications are BRDC, severe foot rot, and diphtheria, bacterial enteritis, wooden tongue, leptospirosis, wound infections, acute metritis, and anaplasmosis, and anthrax. Feed and water additive, we would use an anti-coccidial, mycoplasma, and others too. For oral absorption, it's good in small animals for these, but it's poor in horses and ruminants. We still use it orally in horses because we can give a high enough dose so it can be used for therapy reasons for therapeutic concentrations. It can be erratic. Chelation, candidates in the stomach can bind to the drugs, should be given on an empty stomach, and doxycycline is less effective. They distribute well to most tissues, the exception being the CNS, the central nervous system, and the eye. An exception to this is minocycline. It's more lipophilic. Doxycycline also lipophilic, but has higher plasma protein binding. It's primarily limited by the kidneys. The exception being doxycycline is excreted through the bile and kidneys, and minocycline is primarily excreted through the bile. More adverse effects, so skeletal effects, we have dental discoloration, inhibition of lung bone growth, young animals, offspring, and pregnant animals treated with tetracyclines. Doxycycline is less likely to cause this effect. We have hypersensitivity and fevers, particularly in cats, and photosensitization, which are particular with doxycycline. So important adverse effects, we have the esophageal structures in cats, so cats can control their own esophagus. The doxycycline tablets or capsules get stuck there, so they have direct mucosal ulceration within their esophagus and stricture, especially when broken. So you want to give, when you give this drug, you want to give a small amount of water so it actually goes down. We have rapid IV administration of tetracyclines, so hypertension and collapse in any species. We have calcium chelation, propylene glycol vehicles, and IV administration of doxycycline to horses is actually very fatal. They will die. Don't do this. And for nephrotoxicity is rare, but high doses of oxytetracycline only formations with propylyl glycol. It's not. So then we have hepatotoxicity, which is also very rare. We have idiosyncratic toxic hepatitis in humans, particularly in pregnant women, and also in fowls. We have GI toxicity. This is also not very common, but horses, it's rare around here. It's common in Florida, and dogs and cats can vomit. It's resistant. Resistant is widespread among staphylococci, streptococci, and gram-negative enteric bacteria. Enterococci are not susceptible. Resistant is plasmid-mediated, so failure of the active transport system necessary to penetrate the bacterial cell wall. Non-bacterial uses is treatment of heartworms, so dogs and cats. Remember the Wolbachia is a symbiosis with this. So bacterial endosymbiosis has anti-inflammatory effects. It inhibits MMPs. It's typical in eye for melting ulcers and systemically for synovitis and laminitis in horses. So that was it for pharmacology today. And then now I'm going to get into Virology 7, which again, we're on RNA viruses for this. Today we're talking about influenza and also about coronavirus. So orthomyoxiviridae has to do with all our flu viruses. It has a segmented genome. It replicates in the nucleus. There's no inclusion bodies. It has to do with swine influenza, equine influenza, canine influenza, avian influenza, and human influenza. So reassortment. Reassortment can occur randomly in RNA viruses with segmented genomes, such as orthomyoxiviridae, reoviruses, and bunny aviaroviruses. This mixing of various segments of a segmented genome when a cell is infected with multiple viruses results in a rapid antigenic shift in the epidemiology of the influenza virus. It affects the same cell in the animal, so a mix of red and blue have blue and red surface proteins. So the genome stematics come from red and blue. The progeny would be a mixture of red and blue. This is what I'm talking about. So you have a red virus here, you have a blue virus. They come together, and then they create a mixed virus. So that is how viruses get different strains as well. So the orthomyoxiviridae is an envelope which is derived from host cell membrane lipids, contains glycosylated and non-glycosylated viral proteins. Surface projections of glycoproteins form the spikes or the papillomers, which is influenza A and B viruses, and are of two types. So we have a hemagglutinin, which is known as H, causes red blood cells to stick together and is responsible for virus attachment and envelope fusion. And then we have neuraminidase, which is N, which is capable of cleaving viral receptors, degrading mucus, and promoting both entry of virions into cells and the release of virions from infected cells. Influenza A viruses are grouped based on their H and N type. So 18H, H's are also club-shaped, and whereas N's are other-shaped. So 18H and 11N antigens are currently known, with new subtypes emerging periodically. Point mutations result in new strains. So this is known as antigenic drift, and then we have reassortments, which means switching genome segments, which is known as antigenic shift. So then we have the classification of isolate and based on the type, host, location, strain number, year of isolation, and subtype. We have reservoir and host of influenza, so we're going to talk about just type A influenza because it has to do with multiple species of wild and domestic birds, swine, horses, and humans. So I'm going to talk about the cycle of transmission. So the bird strain doesn't replicate well in people, and the human strain doesn't replicate well in birds. However, both replicate well in swine, so in pigs. This causing reassortments. So pigs are a reassortment host for flu viruses, causing a new strain of influenza. So in 2009, a new pandemic strain of H1N1 influenza A began circulating in humans. It appeared in Mexico and rapidly spread. Sequencing studies indicated similarities with the 6 gene segments of North American triple reassortment swine influenza isolates, so H1N2 and H3N2, so called because they contain genes for avian, human, and swine influenza viruses. So getting into avian influenza, aquatic birds, especially ducks, are the reservoirs of influenza A. They provide a genetic pool for the generation of new subtypes. Migratory waterfowl and poultry trade are the cause of the international border spread. Most viruses are species-specific, but transfer between species does occur. Viruses replicate in the gastrointestinal tract of birds, and the transmission of low pathogenic subtypes is fecal-oral transmission. Human infection with avian influenzas is attributed to poor hygiene and close contact with poultry and pigs. Genetic reassortment in pigs and poultry can lead to new subtypes that can infect humans and result in pandemics. Avian viruses replicate poorly in humans, like I said, but human and avian viruses replicate well in pigs, this leading to the reassortment and emergence of new viruses, like I said. Some viruses can infect other species without reassorting, but no human-to-human transmission has been documented. So highly pathogenic avian influenza is caused by influenza A subtypes H5 and H7. High pathogenicity H5N1 in Asia. High morbidity and mortality in domestic poultry, 170 cases and 92 deaths in humans. Direct transmission from poultry to humans. Human-to-human transmission is inefficient. So I have avian influenza in the U.S. So December of 2014, several cases of high pathogenic avian influenza were reported in the backyard flocks along the Pacific Flyway. So April 2015, the first reports of HPAI in commercial flocks came in Minnesota and Wisconsin. By May of 2015, the virus has spread to Iowa and South Dakota. The last detection was reported in June of 2015 after 50,400,000 birds were affected in 15 states, resulting in a loss of $3.3 billion. January of 2016, HPAI was reported in Indiana, but was contained to one flock and was shown to be a mutation of low pathogenic avian influenza. So HPAI is high pathogenic avian influenza. March 2017, HPAI was reported along the Mississippi Flyway. In 2020, LPAI was identified in North Carolina. It spread to South Carolina and then mutated to high pathogenic avian influenza. So apparent risk factors for the spread of high pathogenic avian influenza is contact between wild and domestic birds, live bird markets spread amongst domestic birds, close contact between humans and infected poultry. We have equine influenza, so outbreaks are associated with shows, sales, racing, and or training. Horses 1 to 5 years old tend to be most susceptible. Clinical signs include cough, high fever, and nasal discharge. Transmission is by aerosol or contact with contaminated equipment. The virus replicates in epithelial cells in the upper respiratory tract. This yields rhinitis and tracheitis. Typically H7N7 believed to be extinct now, but is now H3N8, respond by isolating that animal and people wearing PPE so it doesn't spread. Swine influenza, the severity of disease depends on the subtype H and N. Clinical signs include cough, high fever, nasal discharge, and occasionally abortion. Transmission is through direct contact or aerosols, so these viruses don't live well in the environment, so it's easy to disinfect. The virus replicates in epithelial cells in the upper respiratory tract, resulting in rhinitis and tracheitis. Subtypes are typically H1N1, H1N2, H3N2. And then we have canine influenza. This is H3N8, so equine lineage, but was first reported in Florida in 2004 in racing greyhounds. It has then spread to 40 states and Washington, D.C. It is now considered endemic in Colorado, Florida, New York, and Pennsylvania. H3N2, the avian lineage, has caused a large outbreak in dogs in the U.S. in 2014 and 2015, which then spread to cats. Mutation in these viruses allowed for dog-to-dog spread. The virus is closely related to the Asian strains identified in 2006. The first cases in Canada were in 2018 and in 2 dogs from South Korea. Transmission is through aerosol and contaminated equipment. Dogs are most infectious during the 2 to 4 day incubation period before the onset of clinical signs. Virus can shed before clinical signs are seen. This is a major problem. The virus replicates in the epithelium of the respiratory tract, including nasal mucosa and bronchioles in the lungs. Infection results in rhinitis, tracheitis, bronchitis, and bronchiolitis. Necrosis of the epithelium can predispose to secondary bacterial infections. Clinical signs include cough, high fever, and nasal discharge. So comparing and contrasting, influenza virus replicate in the epithelial cells of the respiratory tract. Infection results in rhinitis, tracheitis, bronchitis, bronchiolitis in dogs. Clinical signs include nasal discharge, high fever, and cough. Transmission by aerosol, direct contact, or contaminating equipment. Swine influenza can cause abortion. Wild birds are the reservoir of most viruses and provide the genetic material for reassortment and emergence of new subtypes. So pigs being the reassortment host. Then we have copy choice template switching recombination. So coronaviruses. So now we're heading into coronaviruses. Coronaviruses cause these 3 things. So they cause GI disease, respiratory disease, or neurological disease. They can go all different routes. Commonly it occurs between positive sense signal stranded RNA viruses. The RNA polymerase switches between template strands during synthesis of the complementary negative sense strand. An exceptionally high frequency of genetic recombination has been observed in mixed infections of coronaviruses. So they're in diameter, they're 75 to 106 nanometers long, their envelope, their symmetry is helical, their transcriptase is negative, and they're single-stranded positive sense. Key context of the coronavirus, their host cell tropism is typically respiratory intestinal epithelium. Infection is usually mild in older animals, but can be severe in young animals. This is the opposite of COVID, however, so that's the exception to the rule. Coronaviruses are associated with important diseases in animals. Alpha coronavirus 1, so feline coronavirus, including feline infectious peritonitis virus. transmissible gastroenteritis virus, and canine coronavirus. Porcine epidemic diarrhea virus and beta coronavirus 1 includes bovine coronavirus, equine coronavirus, and canine respiratory coronavirus. So we're going to go with the feline enteric coronavirus, so the feline infectious peritonitis virus, and its life cycle. So it's actually very long and complicated. It can have many. It can actually have 3. The feline enterovirus shed in the feces of a carrier cat, thus infection in kittens, so replication in enterocytes. This leading to the subclinical or clinical infection, or it leads to the recombination or mutation of spike or 3C protein, which causes protective of infection of monocytes and macrophages, which leads to the feline infectious peritoneal virus. However, if it goes to the clinical or subclinical infection way, this leads to adequate mucosal and cell-mediated immunity. So this leads to the virus being eliminated and a clinically normal cat. However, if the feline infectious peritonitis virus yields, or it becomes this, so we then have the replication of macrophages, this leading to many pathways it could take. So after the replication of macrophages, it can go to inadequate cell-mediated immunity, so intermittent replication of FIPV. Clinically, so then you get a clinically normal cat, but it has periodic shedding of the virus. Or if the intermittent replication yields immune complex formation in the circulation, this leads to immune-mediated vasculitis, so paravascular clusters of virus infector macrophages, severe or moderate. Severe being, it yields to marked vascular permeability, which leads to effusive wet FIP and progressive fatal disease within weeks. If it's moderate, this leads to granulomatous lesions, so it's non-effusive dry FIP. However, it still leads to progressive fatal disease in months. Or it could take a whole nother route. So from the replication in macrophages, it could yield ineffective cell-mediated immunity, so the cells cannot detect cells to kill them. The continuous replication of FIPV, so the immune complex formation in circulation, which yields immune-mediated vasculitis, can lead to severe or moderate, which was the same as above. Or the third route, the replication of macrophages can yield B cell activation, production of non-protective antibodies, which yields the immune complex formation in circulation, which yields the immune-mediated vasculitis, which leads to severe or moderate, all leading to the death of FIP. So the key initiating pathogen event in FIP is the productive infection of macrophages and monocytes by genetic variants of the original enteric coronavirus. So what is considered the most important differentiating factor that is associated with one cat exhibiting the wet form of FIP and another cat exhibiting the dry form of FIP? Well, that's the severity of the immune-mediated vasculitis. And then a safe and highly effective vaccine is available for FIP. That is false. There is only one licensed FIP vaccine available. However, the vaccine has minimal, if any, effectiveness in preventing FIP. It's not generally recognized by the American Association of Feline Practitioners Feline Vaccine Advisory Panel. Permissible FIP, previously produced by Pfizer Animal Health, was a temperature-sensitive modified live virus vaccine given as an intranasal vaccine licensed for use in the cats at least 16 weeks of age. The vaccine appeared to be safe, but the risks and benefits of vaccination were questioned. So then we have canine coronavirus, which is the ability of the canine coronavirus to cause disease is variable, and the agent can be isolated from normal dogs to those with diarrhea. 2 genetic types have been recognized. That's the canine coronavirus type 1 and canine coronavirus type 2. A genetically and antigenically distinct virus is the canine respiratory coronavirus. It has been described and is associated with mild respiratory disease, particularly in kennel dogs. Then we have transmissible gastroenteritis, which is the severe diarrhea in pigs with almost 100% mortality. There was one stereotype of this, which is closely related to antigenetically to feline coronavirus and canine coronavirus, and all 3 viruses are now considered a single species and referred to as alpha coronavirus 1. We also have porcine epidemic diarrhea virus, and then we have bovine coronavirus, which is one of the causes of calf diarrhea and is also associated with winter dysentery in adult house cattle. The average age of diarrhea onset is 7 to 10 days. There's evidence of its involvement in the bovine respiratory disease complex, and the virus is transmitted by fecal-oral route. We also have equine coronavirus, also known as beta coronavirus 1. It's usually subclinical or self-limiting, typically associated with enteric disease in fowls. In 2010 cases were seen in adult horses. Like bovine coronavirus, disease is seen more in winter months in stressed animals or under poor environmental conditions. Equine coronavirus has not been associated with respiratory disease. So yeah, that was the end of our lectures today. I have to get ready now, 12:15, so I have 45 minutes to prepare for my client simulation. So I have to fix my eyebrows and put on a white lab coat and kind of look like a doctor. But I hope you guys are having a good day, and I will talk to you guys tomorrow.